Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus, and then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. course, in real life, that takes days, about 5 days. At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnant